In today's lesson, let's talk about viruses and prokaryotes. The question of whether viruses are living things is always debatable. At first, scientists believe that viruses are living things because they use nucleic acid, they use DNA and RNA, and that is the same as what other living organisms do to store their genetic information. And another evidence that made scientists believe that viruses were living organisms was that viruses adapt to changing conditions like living things by genetically mutating. However, as scientists discover more and more about viruses, they kind of agree viruses are considered non-living organisms, and that is because they found that viruses are not made of cells and they cannot reproduce on their own. Let's recall from Grayton biology that all living organisms must be made of one or more than one cell, because cells are the most basic units of life. And also, living organisms have to produce offspring on their own in order to ensure the survival of their species, either sexually or asexually. However, like I just said, viruses cannot reproduce on their own. They need to use the cell of a host organism to make copies of themselves. And in the following slide, I'm going to explain how the reproduction happens in viruses. But now let's take a look at the virus's structure. So here I've got some diagrams for you, and the diagrams on the left are the simplified diagrams for viruses. And the diagrams on the right hand side are how they actually look like. And in my opinion, the T4 bacteriophage is the easiest one to distinguish because of its spider-like shape. For tobacco mosaic virus, you can either call it rod-like shape or noodle shape if you think that looks like noodle. Well, for an influenza virus, it has a shape of a ball. And if you examine the diagram, the simplified diagram here carefully, you can see that viruses normally contain three main components, which are genetic material, protein code, and envelope. The genetic material of virus can be either DNA or RNA, and that contains the instructions for virus replication and protein synthesis. And all the virus contain protein codes, or in a more biological way we call them capsid, which protect the viral genetic material. They just kind of wrap around the material of the virus so that they won't get killed easily. Well, for certain viruses, they have a membrane which helps the virus invade the host immune system and aids in the host cell entry. And now let's move on to the virus reproduction. And this is a diagram that illustrates how viruses reproduce. And as you can see, when a virus infiltrates a cell, it uploads its genetic information. It inserts the genetic information to the host cell. And what's going to do next is that the virus will take control of the cell's processes. And that is like when you accidentally download a virus into your computer and the hacker can access your personal information such as your bank account or your email addresses. The viruses that I'm talking about here cannot do that. They are the actual virus. But what they can do is that they can take control of the whole cell processes and then they'll replicate. And the replication may happen immediately or wait for a period of time before any changes occur to the cell. And you might be wondering how they insert the DNA into the whole cell. And there is actually a very complicated procedures or process behind it, and I'm not gonna talk too much about it, but for the ones who are interested in, this process is usually done using the tail fibers or the spikes of the phage, like the end of the virus. and they will bind to molecules like proteins and other substances which I'm not going to mention here on the bacterial membrane. And once they are attached to each other, the phage will contract its tail sheath like a syringe, and therefore the DNA or the genetic material of the bacteria, sorry, of the virus will be inserted into the host cell. And for the ones who do not know what a syringe is, here I've got a picture of syringe for you. So there are actually two different replication cycles, but they're similar in essence. And what do I mean by that? You can actually answer the question by looking at a diagram here. As you can see, after the last phase in the lysogenic cycle, the virus will enter the third phase, or the second last phase in the lytic cycle. Now let's figure out the similarities and differences between the lytic cycle and the lysogenic cycle together. So in the lytic cycle, the bacteriophage attaches to the host cell and injects its DNA. And this forces the host cell's enzyme and synthesis machinery to make numerous copies of the viral DNA and viral proteins. And the viral proteins and the nucleic acids then assemble themselves inside of the host cell, making many copies of the original infecting virus. And once the number of the virus are anywhere from 20 to 500, the number really depends on the size of the virus or the size of the host cell. The host cell can no longer hold them anymore and it will burst open. Or in a more scientific way, it's called cell lysis. And in terms of the lysogenic cycle, the viral genes are incorporated. Okay, so the first step or the first phase is exactly the same as the lytic cycle. And the next step starts to differ. In the lysogenic cycle, the viral genes are incorporated within the host cell's DNA. They're like spies or imposters, and the host cell does not realize that it's already getting infected by the virus. So it starts to replicate, and therefore, the genetic material of the virus will be passed along the daughter cells of the original cell. And the change in temperature or the change in pH level or other factors can trigger the viral DNA to separate from the host DNA and start a lytic cycle, which means in certain conditions, the virus will enter the third phase in the lytic cycle. And now you might be wondering when viruses enter lytic cycle and when do they enter lysogenic cycle? Well, pause the video and think about that for a minute. I hope you had a go. Well, the answer is that normally the virus will choose a lytic cycle over a lysogenic cycle because apparently it's a more efficient, faster for them to reproduce through lytic cycle. It saves their tons of time. And why do they still sometimes choose lysogenic cycle? And the reason is that they will choose lysogenic cycle under extreme conditions, like the temperature is too high or the pH level is too low or other factors. So when the environment is good for them to survive, they will just get out of the cycle. They will just get out of the lysogenic cycle and they will enter this phase of the lytic cycle. And now let's review some key terminologies in virus reproduction processes. Well, the first term we're going to be looking at is latency. Well, I did not really mention the word previously, and I apologize for that, but it basically means the ability of the virus to lay dormant within a cell. So latency happens specifically in the lysogenic cycle. And the second terminology is bacteriophage, which means a virus that specifically infects bacteria. And you can actually guess the meaning of the word from the prefix bacterial. So that is definitely bacteria related word, right? The third term, lytic cycle. And that is just a normal process of viral production. Well, lysogenic cycle happens under certain conditions. well, let's just end it here. And originally, I wanted to talk about viruses and prokaryotes in one video, but the thing is that it seems like no one really want to watch a 20 minutes video explaining some dry and boring grade 11 biology content, and I understand that. So I decided to put them in two different videos separately in order to make each one shorter. And anyways, thank you for listening, and I'll see you in the next episode.